I'm Jackie Cohen-Roth, founder and CEO of Cannabis MD. Cannabis MD is focused on filling the void of cannabis science and policy education, and we are accomplishing that via conversations that I've been having with medical cannabis subject matter experts. I am very excited today to be joined by Dr. Nandu Sarma, who will be one of the subject matter experts on a webinar January 19th that we're hosting, beginning at 4 p.m. EST time. And that conversation will focus on the need for cannabis, the standardization of cannabis science education for physicians. So welcome, Dr. Sarma. Delighted to have you join.
Pleasure to be here, Jackie. Thank you for having me.
Thank you. So Dr. Sarma, Nandu, thank you so much, is director for the Dietary Supplements and Herbal Medicine Program at US Pharmacopeia, known as USP. He's responsible for strategy and external stakeholder engagement for new and innovative projects, working with global stakeholders and expert volunteers in the development of quality standards for dietary supplements and herbal medicines that are published in the USP Dietary Supplements Compendium and the Herbal Medicine Compendium. Dr. Sarma is a pharmacognosist, if I can pronounce that this morning, by training. Before joining USP in 2006, he was a postdoctoral fellow at the National Cancer Institute, NIH, in Bethesda, Maryland, and Thomas Jefferson University in Philadelphia, and was a senior scientific officer at the Himalaya Drug Company in India. His research experience includes isolation and analysis of active components of botanicals and their biologic activity. He has published more than 25 scientific articles in peer-reviewed journals, and he holds a pharmacist degree and a PhD in pharmaceutical science from Banaras. Am I pronouncing that?
Banaras.
Banaras, Hindu University in India. So excited to get going with you. So let's begin with what exactly does it mean to be a pharmacognosy? And again, I'm not able to pronounce it this morning.
Yes, so yeah, that's an interesting start for the conversation. For those who are not very familiar with the term pharmacognosy, let me define it.
Thank you.
It's a study of natural products associated with some of the medicinal or biological activities. So mainly they consist of botanicals and even something like marine organisms are also part of the natural products, which could be within the scope of pharmacognosy. And the study involves study of physical characteristics, like in case of botanicals, their identification using how it looks, what's the morphological features, what is the microscopical features, and all the associated topics such as their cultivation, collection, post-harvest practices. So it's a knowledge about the botanicals. And beyond identification, the study also involves the phytochemical profile of a botanical ingredient, so that it can be appropriately fingerprinted, it can be differentiated. There's a lot of biological diversity, so you may have many plants which look alike, and they might also be called with similarly sounding names. So it's important to differentiate it appropriately and name them appropriately. So it's a lot of science around it. Beyond identification and characterization, biological assays, biological studies, including pharmacological activities on these. So the objective is to evaluate their beneficial effects as botanical drugs or botanical dietary supplements. So the science is gaining more attention pertinent to our conversation. Cannabis is indeed bringing up more awareness about the pharmacognosy as an area. And what it means to be a pharmacognosist, both for me and for others, is to be a subject matter expert in this area and also to share the knowledge and passion with others.
Yeah, well, thank you for that. And for some background on the relationship of cannabis with USP, cannabis was used by physicians for therapeutic benefit from the
Mhm.
19th century and made its way to the U.S. Pharmacopeia in 1851. It became illegal via the Tax Act, Marijuana Tax Act of 1937 in the U.S. And then subsequent to that, it was dropped by a U.S. Pharmacopeia in 1942. And then under the Controlled Substances Act, the CSA of 1970 became federally illegal. So today, what is the approach with USP and specific to our conversation, cannabis?
Sure, glad to elaborate on that. Let me take a step back and
Mhm.
share USP's connection with botanicals over the last 200 years. We recently celebrated 200-year anniversary. USP was founded in 1820 and has
Wow.
been working on botanical quality standards since then. And I will share a copy of our publication in the Herbal on the USP's contributions to botanical quality standards over the last 200 years. And in it, we described cannabis as one of the, from the historical perspective. So we had a monograph in USP since 1850 until 1942. And in fact, one novel thing about the USP monograph in the USP edition 9 was that it contained bioassay for cannabis. So it was a intoxication endpoint in dogs as a way to measure the potency. So it was interesting, even in those days,
Yeah,
there was biological relevance
yeah.
for standardization. So that was a historical background, and apparently USB has admission criteria for admitting or deleting or omitting a monograph, and it was omitted in 1942. And USB has received lots of representations that there is a need for public quality standards in this area. And in 2016, USB organized a roundtable discussion on the advisability and feasibility of developing quality standards for cannabis, and also wrote a stimuli article on this, elaborating on this topic. Based on those conversations, USP formed a expert panel, the cannabis expert panel, with subject matter experts, volunteer experts from different subject areas, including academia, industry, regulatory affairs, and testing laboratories to advise on the quality standards. And um, most recently, in 2020, USP published the work in the Journal of Natural Products.
Hmm.
So the first publication was on the cannabis flower, and a monograph was not developed because of the federal status in the United States. It is regulated differently in different countries and in different states within United States. But the federal regulatory status is important for USP because USP is an official compendium in the Federal Food Drugs Cosmetics Act. So there are implications associated with admission of a monograph into the USB. But in order to meet the public health need, USP followed the same process as we follow for other botanical drugs or any other prescription drug, using the expert volunteer process, a transparent process involving a science-based approach. So the paper was provided to provide these quality attributes for identity, purity, and the limits on contaminants. So this is a very brief background of USP's contributions in this area.
Yeah, and one particular contribution ongoing right now is a discussion on the minor cannabinoid that's getting a great deal of attention now, delta 8 THC, which was doing my research while in grad school, that it goes back to 1970, what I had found in actually Maryland, where I'm based, where a couple of folks extracted and processed delta 8 from hemp and using petroleum. And with that, they developed delta 8, and which the dangers that I've been letting folks know, whether on with Cannabis MD, social media platforms, my own platforms, is that for folks to realize that the dangers that are involved, and that there aren't that many lab tested delta 8 products out there. So if you would provide a background on delta 8 and how USP has been working with ASTM International on educating folks in the cannabis industry, healthcare providers, as well as consumers on the dangers of delta 8.
Sure. If you see the USP publication in the Journal of Natural Products, we have recognized that delta 8 is one of the minor cannabinoids, the major ones being the CBD, CBDA, THC, THCA. And after review of a large amount of data, USP noted in the publication that the amount of delta 8 present in the cannabis, including hemp, is very low to insignificant. It was present in highly oxidized samples as a degradation marker, to put it that way. But you don't really get a lot of delta 8 in the plant by itself. Another important observation in the paper was the lack of research and information on these minor cannabinoids. So it was identified as one of the major research gap in this area. Mm-hmm. So that was an observation very early on. The paper also provided analytical methods to resolve or differentiate between the delta 8 and delta 9. So, when you have so many different cannabinoids in the cannabis plant, they can co-elude with one another. So you may either underestimate or overestimate. If something is not visible, if it is hiding in like delta 9, you might overestimate delta 9 and you might underestimate delta 8. So we need appropriate methods. And in fact, in the paper, the resolution of the delta 8 and delta 9, because it is so close, is included as a system suitability requirement to show that your system performs appropriately. So these are all were highlighted in the paper. And now the recent observations are about synthesized delta 8. So what you alluded to earlier was about extraction of the delta 8 from hemp material using petroleum or petroleum ether as a solvent. What we observed more recently was the conversion of the CBD into delta 8 through synthetic process. So there are there is a very well-known patent from Dr. Mechelen available that gives a lot of different processes. And normally the process involves use of harsh conditions such as strong acids, catalysts, and purification. And depending on the method, it might be accompanied with different degradants, reaction intermediates, or other cannabinoids which are not present in the plant naturally, because you're conducting a reaction from a cannabinoid to a different one, and it's not a one-to-one conversion. Mm-hmm. So the expert panel had discussed this topic and observed data of many unknown cannabinoids, unknown compounds that accompanied the the Delta 8 products in the market and provided a commentary on the concern about concern about the quality of these products, the lack of research, and made some recommendations to reduce risk of harm from these products. So it's not a commentary on the inherent safety of the Delta 8 or the process itself, but it is rather the quality concerns and the lack of research. So it's not to be taken as a condemnation of Delta 8 or condemnation of the synthetic process in general. What is needed is a systematic study to understand at what level it is safe, effective, and using very well quality controlled material. So that's where the quality, public quality standards can help in that regard. And FDA has provided guidelines for assessing the quality of cannabis for clinical studies. So there are paradigms available for evaluating these. And we also worked with ASTM. They were wonderful partners who shared the similar concern. And ASTM also came out with a commentary or a white paper on this topic. And so USP's focus was on the quality aspects. ASTM was apparently much broader, going even into the details about how the retail industry could help using some of these recommendations and so on. So it was a common public health concern for which USP and ASTM provided commentaries recently.
Yeah, and thank you. And to put that into context is where Delta 8 has is so prevalent and available, and as you spoke to the needs for safety standard, is that as a result of the 2018 Farm Bill, that hemp became legal, has become legal off of Schedule 1. And
it is descheduled, yes.
Descheduled, thank you. And so, as I say, everybody and their grandmother got into the hemp industry, and there was this overabundance of hemp. And so this is, what do you do with that biomass? And so this was, as many of the those who have less strict adherence to safety regulations saw this and see this as a huge money-making opportunity. And where would you like to buy your cannabis products? At either a dispensary where products have gone through these necessary standards, testing standards that we're speaking of, or at your gas station where who knows what products have been used to extract and develop this Delta 8. So that's
indeed, yes.
Yeah. Okay. All right. And then let's move on to the topic, the focus of our discussion on January 19th, which is what I consider the need, as do many, for standardization of cannabis science for healthcare providers. And I regularly use my data point of coming from a 2017 Wash U study that identified only 9% of medical schools include cannabis science in their curriculum, whereas now the majority of, speaking only to the U.S. right now, the majority of of Americans have access to THC products. There are now over 50 countries globally that have some sort of a medical cannabis program. So what is USP doing in the role to help standardize, or what do you see as specific standardization needs in this arena of cannabis science education, and again specific to physicians of what we're talking about?
Yeah, so let me take one step back
Sure.
to 1820
That's
when
a big
11
step.
yes. So 11 physicians met in the Library of Congress to develop the U.S. Pharmacopeia, and the concern at the time was that you travel across the different states, and even within the state, different areas. There was concern that, for the same name, you would get a different product. So you don't have that certainty of the quality. Uh, the fundamental aspect was, connecting the name with the quality was not there. For example, valerian was in USP 1820, and it is used as a dietary supplement even today. So the contribution at the time was to provide a simple recipe of how to make it, so that anywhere in the country, if you call it valerian tincture, you get the valerian tincture meeting certain quality specifications. So in my personal view, the situation is not very different today with respect to the cannabis. So and more specifically about cannabis, there are so many varieties of the cannabis plant.
Right.
People recognize that, and people recognize with the different names of what they call strains of cannabis. So the strains is a misnomer. There is not, it's not a botanical term. It's a variety, the cannabis varieties. And the varieties are broadly, it's a CBD dominant variety, THC dominant variety, or the intermediate variety. There are genetics that play into how things could be, how the varieties could be evolved to produce certain cannabinoids and not the others. So if you were to, or if a physician were to make a recommendation that, okay, give him cannabis, really doesn't convey what the intent is. So what USP's contribution in this area is to provide a standardized nomenclature. So if you see the USP publication, you would appreciate the work starting with the nomenclature so that it is named appropriately.
And we're talking as, and I see the challenges of how are we going to add and remove or correct myself, remove the stigma for healthcare providers as for them to, at someday, right now they're writing recommendations, someday to write a prescription for something called Granddaddy Perp or Blue Dream is that, speaking of nomenclature.
That's right. That's right.
Yeah.
And these terms, these names could mean different to different people.
Right. Yeah.
And so what is needed is the intent of what the physician intended to address in the patient, and what particular variety of the cannabis is needed with specific quality attributes to address that particular need. So starting with the nomenclature and connecting the name with a specific well-defined acceptance criteria. So if you say, there are certain conditions for which CBD dominant variety is recommended. So what is the content of the cannabinoid that is expected, and what should be, you don't want to get high, you need to control the THC. So what is the how do you control that? The name is connected with the quality specifications, and quality specifications in terms of the identity to make sure that you can differentiate these different cannabis varieties, not just by the smell that doesn't really convey anything. It is a organoleptic sensory perception, but that doesn't really communicate whether it is what it is. And with a specific acceptance criteria, that if you call this variety, then it has to have these criteria, which are unique for that particular name and can differentiate with other varieties, and also provide analytical methods that can appropriately be conducted to evaluate that variety, and an acceptance criteria that can say pass or fail. It is binary, so whether it meets those criteria or not. And associated with recommendations about the labeling as to what is the content of the total THC, what's the content of the total CBD, what's the what's the limit for the degradation components, such as CBN could be a degraded, a marker for stability, marker for shelf life, and a expectation that the product is stable and maintains the quality all through the shelf life. So this is an area where USP brings its strength in terms of providing these inputs, so that the risk of harm to the patient from substandard, adulterated, contaminated products is avoided. The specifications also include a very elaborate description of how to limit these contaminants, such as pesticide residues, microbial load, residual solvents, heavy metals, and things like aflatoxins. So that's a unique contribution that USP brings. And USP is a public health organization. The objective is to reduce risk and to promote research. So these are the twin objectives, which were the drivers for USP's work and contributions in this area.
How about parallels in the global markets of organizations? There are particular countries that you have professional relationships with that you see as setting these sort of standards?
Yes, yeah, we do have conversations with regulators in different regions, and the challenges are very similar. The challenges um, that we observe with the different state requirements within the United States are not dissimilar to the challenges that different countries are facing. So each country has its own law of land to implement. The requirements are slightly different. For example, in some cases, the hemp is defined as containing not more than 0.2% of the THC,
Right.
or maybe in some cases, maybe 1% of the THC is permitted. So the requirements could be slightly different, but the challenges are very similar.
Yeah, my understanding is that within the EU, there's a far greater consensus of GMP versus looking at in the US, where every state seemingly has a different model. And this is where the industry needs to collectively come together and create GMP.
Yeah, so the ASTM is doing a good work in this area to write these quality management systems and providing guidelines all the way from seed to the shelf. Lots of different areas that need control. So GMPs are the broader quality systems. And because cannabis is federally Schedule 1 substance, there are no GMPs that are applicable all over the country. However, there are certain good practices. For example, the 21 CFR 111, the GMPs for dietary supplements, is applicable for botanical dietary supplements. So some of those good practices could be adopted here, and that's what we see people doing. And of course, if and when cannabis becomes a drug, it is subject to the GMPs applicable to the drugs in the same way as other botanicals such as the green tea extract and the dragon's blood are subjected to. Cannabis would also be subjected in the same way.
All right. Well, thank you so much for your time. It's just fascinating conversation, and with his 200-year history.
Indeed.
Yes. If our audience would like to learn more about USP and specific to cannabis, where should folks look to?
So we have a well-populated landing page. So it is www.usp.org/cannabis. So all the information we post in one location. So that's a good place to start.
Okay. Well, thank you so much, and look forward to our discussion on January 19th with medical cannabis industry colleagues. And
My
thank you
pleasure.
again, Nandu.